Welcome back, ladies and gentlemen. We are now about to commence the academic program. And to commence the first plenary on next generation insulins, let me invite the two moderators, Dr. Udita Bulugahapitiya, consultant endocrinologist from Colombo South Teaching Hospital, Sri Lanka, and Dr. Charles Anthony Pillay, consultant endocrinologist from Teaching Hospital, Kandy, who is joining us virtually. Over to you, sir.
Good morning, ladies and gentlemen. It's my great pleasure to welcome all of you to the first plenary of this International Diabetes Update by SAFES. To speak to us, it is Professor Anne Dornhorst from the United Kingdom. She's a consultant physician and honorary senior lecturer in endocrinology and diabetes at Imperial College Hospital, London. Over to you, Professor Dornhorst.
Okay, I'd like to thank SAFES for the invitation to speak to you this morning. And I've been asked to speak about the new generation insulins, a glimmer of hope for South Asia. Now, I'm going to cover very briefly the ultra-long insulins, the ultra-fast insulin, high-strength insulins and what they have to offer, biosimilars and smart insulins. That is now 98 years since the first introduction of bovine insulin, and we've come a long way. And since the 2000, when was the first introduction of the long-acting insulin analog glargine, or Lantus, we've had several other developments. We've had another long-acting, ultra-long-acting Degludec. We've had the introduction of high-strength insulins, insulin glargine 300 being the first one. And we've now got very fast-acting insulins, and we have had on the scenes a lot of biosimilar insulins, and they continue to grow. And in the future, we're going to see the very smart insulins. So if we address what is an ideal basal insulin, we are really looking for one with 24-hour plus duration and a flat, predictable profile. So if we look at A, what we have the, it's not flat, there's a peak of insulin over time and then comes down. B is much smoother, the mean, but
Professor Anne, sorry for disturbance. Professor Anne, can you share the screen with us? Share the screen. We can't see your slides properly. Share screen.
Yes, I'm going. Let me just go back to Zoom. I did share the screen. Share.
Oh, that's fine. That's great.
Okay, so I'll just go through them very quickly. The ideal basal insulin would have a 24-hour duration and a flat, predictable profile. So we don't want, if we're injecting insulin here from one day to the other, to have a peak and a lot of variability. We might want one that the mean is okay, but again, there's high variability. What we want is a day-to-day injection that is ideally smooth and a flat mean with low variability. The pharmacokinetics of an insulin depend on its absorption, its distribution, clearance, metabolism, and elimination. And these are influenced by the molecular structure of the insulin, the dose and concentration, the injection site, and temperature. Now, the pharmacodynamics of an insulin are the glucodynamics effect of the insulin. And these are determined, or rather estimated and validated with a euglycemic hyperinsulinemic clamp. So you inject the insulin here, clearly the plasma glucose would go down, but to keep the plasma glucose constant, you have you infuse glucose at the same time, which is known as the glucose infusion rate. And if you measure the plasma insulin of what you've injected, you'll get the pharmacokinetic profile of the insulin by seeing its concentration, while this is giving you the dynamic approach. And this is influenced by the weight and insulin sensitivity. So somebody who's very insulin sensitive would need less glucose infused, would need more glucose infused to maintain a static constant glucose level than somebody who was very insulin resistant. And this is affected by age, health, and physical fitness. Now, NPH was the first basal insulin. And what we have here is we have 9 individuals who have undergone 4 dosing days of a euglycemic clamp. And what we see with, if you inject NPH here and you infuse glucose, that the variability in terms of how long the insulin is working in terms of how much glucose you have to infuse to keep a constant glucose varies from day to day within that one individual. More so in some, less so in others. But this NPH is not a 24-hour basal insulin. There's individual variability, and there's a lot of between individual variability. So it's not predictable, its action. And what one's looking for is one that is predictable, both in terms of its PK and its PD, to reduce glucose variability in subjects with diabetes. Now, the first basal analog insulin that tried to do this was insulin glargine that came in 2000. And it's analog insulin with a substitution in the A chain at 21. And this substitution that also had some arginine substitution in the B chain made it less soluble at a physiological pH, that stabilized the insulin hexamers that aggregate on injection in the depot and slow its overall absorption into the bloodstream. And that so the depot had a longer duration of action than NPH. And insulin glargine would be released following 2 active metabolite stepwise enzymic actions with the removal of the C-terminal arginine. And these would be required to slow before it was absorbed and active. And if one repeats these hyperinsulinemic euglycemic clamps in 9 individuals with type 1 diabetes, what one is seeing is much greater variation between one individual than one saw with NPH. These are much more predictable than with NPH, but there's still variability between one individual and another. But what one also sees, because you're injecting the insulin here, you're infusing the glucose here to maintain a plasma glucose level that is not plotted but would be up here, is that the actual duration of action is not 24 hours. And it's around, 16, 18 to 22 hours in most people, not in all people. In this individual, it's run out by 16 hours one day, but in another day, it's gone on to 22 hours. So it's not predictable. And what one's looking for is something that's much more predictable, not only within one individual, but between individuals. The second basal analog insulin was detamir, that from Novo Nordisk. This is a very clever insulin. Again, it's an analog where they've made a substitution in the B chain of insulin, and they've attached a fatty acid to the end of the B chain. And what this has done is that when it is absorbed, it binds and unbinds and binds again with serum albumin in the interstitial fluid. And this binding gives it a more predictable pharmacokinetic profile and pharmacodynamic profile. And again, if you perform the hyperinsulinemic euglycemic clamps, you can see that the inter-individual variation from one clamp study to another on a different day in one individual is less variable, but it remains variable and unpredictable from one individual to another. And the duration of action of detamir is, again, certainly not 24 hours, and is more like 16 to 18 hours in most people. And so insulin detamir is very much a BD, twice a day insulin in most individuals with type 1 diabetes if they haven't got any endogenous insulin of their own. So the ideal basal insulins would have a 24-hour profile. And the ultra-long insulins are doing this. And we've got two currently on the market. We've got Glargine 300, the high strength insulin. So in 1 ml, you've got 300 units. And you've got the Degludec, 100 units per ml. And it's also got a higher strength one, 200 units per ml. And what one sees with the ultra-long insulin, since they last over 24 hours, you're not getting the effect that you get with Glargine or detamir, that it's out of the system by the time you inject it the next morning or the next night at the same time. But you're getting one that lasts longer than 24 hours. And therefore, after 3 to 4 days, you arrive in steady state. And you've got a insulin action and an insulin concentration that is steady over a period of days and weeks. And there have been several randomized control and real world studies now using these ultra-long acting insulins. And there are certainly advantages in their use. They lower the intra-individual variability in plasma insulin levels. And very importantly, they allow flexible dosing. So you don't have to give it exactly the same time every day. And this has got enormous advantage for shift workers. And it's got advantages for individuals who depend on others to give their injection of insulin, who can't necessarily rely on coming in exactly the same time every day. And all the studies, both the ones done by the pharmaceutical industry who have manufactured these insulins, and in real life observational studies, there's a decrease in the risk of hypoglycemia. And this will come because that they are more predictable and easier to titrate. Now, Glargine 300 is a high strength insulin. So for any given volume of insulin, so if you're giving a 90 units of Glargine or Lantus, I need to go back, forward. If you're giving 90 units of Glargine, that the volume of the high strength insulin is 1/3 of that. And I said before that when Glargine is injected subcutaneously, it undergoes two-step metabolism. Absorption of the high strength insulin is longer. And what one can see in the graphs on the right is that the ordinary Glargine 100 in blue has a shorter duration of action than the Glargine 300. And also that the Glargine 300, and you've got different doses given, that is flatter than that seen for the glargine 100 or the Lantus. So U300 insulin glargine offers a smaller depot surface area leading to a reduced rate of absorption. It provides a flatter and prolonged pharmacokinetic and pharmacodynamic profile and more consistency. It has a half-life of 23 hours and it's within steady state in 4 days. And it has a duration of action that's around, 30 to 36 hours. And you can see here on this graph where you've got different doses of glargine 300 in purple, that you've got a very flat profile compared with ordinary glargine 100 that is here in blue. So the profile results in fewer episodes of hypoglycemia, and a meta-analysis of the addition and free studies showed a similar glycemic control between both glargine formulations across a large and clinically diverse population of people with type 2 when compared with glargine 100. And you can see this on the left-hand side of the screen. And when compared with glargine 100, patients treated with glargine 300 had reduced annualized rates of confirmed or severe hypoglycemia. And this was especially true in terms of nocturnal hypoglycemia. And this study was in individuals with type 2 diabetes. That real-life data outside the randomized clinical trials have also shown that using glargine 300 compared with other basal insulins, that the 6 months after the switch to glargine 300 or another basal insulin, that the HbA1c might be the same, but the amount of patients with hypoglycemia is less, and patients associated with hospitalizations from hypoglycemia is much less. So glargine 300 is a very predictable, good basal insulin, and we certainly use it in the UK for people with type 2 diabetes, because remember that is a high strength insulin, so you've got a third of the volume being injected, and so if you've got highly insulin resistant people who might be on 100 or 90 units, that the actual volume you're injecting is much less. Now, the ideal basal insulin, clinically, the biggest difference that glargine 300 has is it's highly concentrated, making injection volumes a third, like I said. It's an absolute excellent basal insulin for people with type 2 on large volumes of insulin. And also patients requiring flexible dosing times. And this is true, as I said, for shift workers and individuals relying on other people to give their injection of insulin. Now, Insulin Degludec came into circulation after glargine 300, and it's another ultra-long insulin. And like the Novo insulin Detamir, again, it's got a fatty acid substance attached to the end of the B train. And this modification confers a unique property that prolongs its mechanism of absorption into the bloodstream. That insulin degludec has a high affinity binding also with serum albumin, like Detamir does. That, because the only insulin has to be a monomer for it to be active. And what insulin Degludec does, that after absorption, it forms multi-hexamers that take time to break down to dimers and then monomers before it's absorbed into tissues and the bloodstream to become active. And randomized crossover trials of this insulin in patients with type 1 compared with glargine 100 does result in lower rates of overall symptomatic hyperglycemia episodes over 16 weeks and maintenance period in individuals with type 1 diabetes. And again, this is because every day it has a much more predictable action. It's easier, therefore, to titrate than insulin glargine. And it offers much greater flexibility when doses can be taken, as the insulin lasts up to 42 hours, and that allows doses to be taken at different times of the day. Now, we use insulin Degludec, here's insulin Degludec, in vulnerable elderly individuals, mostly with type 1, but they could be type 2, who require district nurses to give the insulin or family members to give the insulin, who can't be relied on to come at the same time every day. And a lot of these elderly, vulnerable individuals require very, very little insulin, but it does need to be predictable. And we use insulin Degludec when often the dose might only be 8 to 12 units of insulin a day, but it really does help. And we often give at the same time something like Humulin-M free that provides some postprandial cover during the daytime and is out of the system by the evening, and allows the district nurses or the family member just to go once a day to give the insulin. And so we give it together, usually with Humulin-M free, or if somebody's very frail and doesn't eat a lot, we might give it with Humulin-I, just to give it a bit of postprandial cover during the daytime, because its profile is so flat. But if you give a sort of MPH like this, that's out of the system by evening time, this sort of area will cover prandial insulin and meal times. It offers great flexibility as doses at different times of the day, and is an excellent basal insulin, as I said, for elderly patients reliant on others to give their insulin. And it really does reduce the risk of hypoglycemia. Now, NovoRapid, or Aspart, that has been modified, that the actual insulin is the same, it's NovoRapid, but they've altered its preparation so it's absorbed more quickly. And I was asked to produce some questions, and the questions are these, and any one is correct. So if you could read them and then decide which is correct. So which of these answers is correct, and around making NovoRapid, which is an analog insulin and is short acting, into a very ultra fast acting insulin. Okay. So the answer was B, that they've added vitamin B3 or nicotinic acid to the formulation of NovoRapid. And what this does is that it makes it much less likely to form dimers and hexamers, and though you'll get more monomers that are the active part of novorapid, and it leads to more rapid absorption of the novorapid into the bloodstream. And so its profile is slightly different from novorapid or aspart, and you get a higher, earlier peak of novorapid. It's not that much more compared with novorapid. And the real place for this ultra-fast insulin probably hasn't met the predictions that Novo had wanted. I mean, is that some people say that we ought to use it in pumps. It's a better insulin for insulin pumps. Occasionally, that individuals who have any form of dumping syndrome, that the insulin ultra-fast acting has got a place. But for most people, we are not using it on a routine basis. Now we come to the biologics, and these really will change clinical practice, and certainly in places like South Asia. So this is the question around the biosimilar insulins. And again, only one is right. Okay. So I think you're meant to decide yourselves. So the biosimilars are a biological medical product, also known as a biologic. They're highly similar to another already approved biological medicine, which is the reference medicine. Biosimilars are approved according to the same standard of pharmacological quality, safety, and efficiency that apply to all biological medicines. Now, what the biologic biosimilars are is that when the patent of the of an insulin runs out, that any individual or any drug company can use that amino acid sequence to make a biosimilar, and that the biosimilars are cheaper, and that NHS England has estimated by using biosimilars that drugs, when they are available, that there's enormous saving in cost for the country. So if you take insulin glargine, which I said earlier was the first long-acting insulin that came in into circulation in 2000, it lost its patent in 2014. And this opened the floodgates for pharmaceutical firms to make a biosimilar equivalent. And this happened very, very quickly. That, as I said, the patent expired in 2015 of Lantus, and FDA approval settlement of a basal came into being 1 year later. And this is a biosimilar to glargine. Its actual amino acid sequence is identical. However, its actual formulation, its 3D structure, its preservatives might be different. But once it's absorbed and it's in the bloodstream, it is for all intents and purposes glargine. And certainly in the UK, it came in at 10% cheaper than ordinary glargine. That we now have Semglee, which is another biosimilar of glargine. Again, the amino acid sequence will be identical, that it didn't have to have randomized controlled trials. It just goes through a very easy licensing process. And in the UK, it's 20% cheaper than glargine Lantus. And there are several other biosimilars that are coming in the coming to the market, being familiar with the older formulations of insulin, and I think as healthcare professionals, we do need to adopt them once they've been around for, let's say, a year or so, and they're proven to be safe, because they are biosimilars. They're not bioequivalent. They're not absolutely identical. And years ago, when a biosimilar of erythropoietin came in, because erythropoietin was very expensive, that there was a biosimilar of erythropoietin that came in that caused, that was antigenically different from erythropoietin because of its 3D structure and its formulation, and in a very small number of people caused aplastic anemia. So they aren't absolutely equivalent, but they are a lot cheaper. There are barriers to using it because everyone's familiar with the older formulations and the names. That also, in our country, we always now use pens rather than syringes and needles. And so you need different pens. So you can't just do a mass change without seeing the individual face-to-face and showing them the new pen device. And there is a reluctance to change. And there have been studies in the UK of what a small percentage of people are actually using the biosimilars of glargine, and the enormous potential loss of saving for the NHS that this costs. So the biosimilars in the pipeline are obviously the ones around glargine, and you've got a Basaglar, Semglee, and Basalin. You've got insulin Lispro, which is Humalog, and you've got a Nova Rapid now biosimilars that will be coming in next year. And again, all of these are marketed en masse, and all of them are cheaper than the older preparations, than the original preparations. And I think that certainly for South Asia, that where cost is important, that the biosimilars have got a lot to offer. Now, the smart insulins are ones that are glucose responsive to insulin. So you have an inactivated insulin, and the switch to make it activated is glucose, ambient glucose, and then it's activated. And there are these in preparation at the moment, and this would obviously be fantastic. I mean, we've got this in part with some of the oral agents, like repaglinide and things, but we obviously haven't got a glucose-sensitive insulin as yet. But these are still very much pipeline insulins. But I think that they will come to be, but they're not quite here yet. And I'm going to finish there. That's a whistle-stop tour of the insulins that have come to be following the introduction of the original bovine insulin 98 years ago. Thank you very much.
Thank you, Professor, for that excellent presentation. And actually, we have a few questions virtually, but I think we have already answered those questions on your presentation. I was very careful listening to your presentation. Therefore, almost all the questions you have already answered. Therefore, I'm not going to put you those questions. And once again, thank you very much, Prof. Zan, for that excellent presentation on behalf of SAFES. Thank you very much.
And thank you for inviting me. It was a pleasure.